All right, welcome back, everybody, to part 2 of our chromosome presentation. So now we're going to talk about something that's called nondisjunction, and nondisjunction is a mistake. Crossing over, that's part of adding variation to a species, and so crossing over is a good thing. Nondisjunction is always a mistake. It's something bad. So take a look at the diagram first before we introduce nondisjunction. Notice that, remember that during metaphase 1 of meiosis, the homologous partners line up side by side. And then in anaphase 1, the homologous partners are separated from each other. So if this is mom's chromosome and this is dad's chromosome, one goes to one side, one goes to the other, and they end up in different gametes. They end up in different sperm or egg. So take a look on the left diagram where it says meiosis 1, and you see the nondisjunction. Two stay stuck together. Instead of the homologous partners being separated from each other, they stay stuck together and they go to the same side of the cell. And so look, as cell division continues, look what that does to the, let's say they're eggs this time. Look what it does to the eggs at the end. The eggs on the left, the first two eggs on the left, all have an extra chromosome. So if those were human eggs, they would have 24 chromosomes. And then the eggs on the right, the next two eggs next to them, they're all missing a chromosome. And so that would give them 22 chromosomes. So that's one type of nondisjunction. There's another type of nondisjunction that happens later in the cell division process. So notice the first time, the first cell, the homologous partners separated from each other just the way they were supposed to. But now we have bivalent chromosomes that are supposed to be separated into monovalent chromosomes. And this time, one of the bivalent chromosomes doesn't rip apart and both copies goes to one side. So then look at the impact it has on the gametes this time and notice the difference. So this time, looking to the right side, the first two gametes on the right, they're normal. If they were human, they would have 23 chromosomes in them. It's the two that are on the left that have the wrong number. So the one on the far left has an extra chromosome, so it has 24 chromosomes, and the one next to it has 22 chromosomes. It's missing a chromosome. So those events are called nondisjunction, and you may remember that we've had this conversation before, that for all species, well, almost all species, it's really important, especially in humans, it's really important to have the correct number of chromosomes. And there's always a syndrome associated when they have the wrong number. So it says, I'll just read this to you quickly. In a nondisjunction event, chromosomes do not separate during either anaphase 1, when they're sitting with their homologous partners and get separated, or during anaphase 2, when they're bivalent chromosomes and need to be separated into monovalent chromosomes. So take a look at the diagram. A mistake in anaphase 1, that's this one over here, leads to 2 gametes with an extra chromosome and 2 gametes that are missing one. A mistake in anaphase 2 leads to 1 gamete that's a +1, it has an extra, and 1 that has a missing chromosome, and then 2 that are normal. All right, so now nondisjunction, it says, must occur in a germ cell as opposed to a somatic cell to have an impact. Now I know I lost my train of thought there. Okay, so what the heck are germ cells? Germ cells, you guys, are cells that determine, cells that are the first cell that divide to make your gametes. So for example, if we saw the very first one of these cells, it would be up here somewhere in the diagram. That's a germ cell. And to help you remember it, I always think G's. The germ cell makes the gametes. Somatic cells are the cells of your body. So this sentence says nondisjunction must occur in a germ cell as opposed to a body cell to have an impact. Why? Well, germ cells are the ones that give rise to the gametes. So if we have the wrong number of chromosomes in sperm or egg and they fuse to make that baby, and then the baby's going through all these cell divisions, every single cell in that baby's body is going to have the wrong number of chromosomes. However, if it happened in, say, my cheek cell, and we got the wrong number of chromosomes there, then it would just be my cheek that's affected, just the cells right in the area, and the rest of my body would have the correct number of chromosomes. So it's much more serious when it happens in our germ cells and when we're making our sperm or egg. Okay, so there are a few genetic disorders that the College Board wants you to be familiar with. So the first thing that we're going to talk about are monosomes and trisomies. This is what occurs when one of the sperm or egg have the wrong number, and we're talking about just a plus or minus one in this case. So let's say we have a sperm that has 23 chromosomes and we have an egg that has 24 chromosomes. When they come together, we get a baby that has the wrong number of chromosomes, and that means that they have three of one of the chromosomes. So instead of having one from mom and one from dad, they got three copies of one. So study this picture. This picture is called a karyotype. It's a photograph of someone's DNA. It's made by a genetic counselor. They photograph the DNA under a microscope, and then they cut and paste using a computer program and move the chromosomes around so that they line up by size. So here's chromosome number 1, and they're roughly the same size. One came from mom, one came from dad. Here's chromosome number 2. There's one there, and there's another chromosome number 2. These are the homologous partners. One came from mom, one came from dad. This is chromosome number 3, and so forth, and they line them up getting smaller and smaller. So down at the bottom, we see chromosome 21, very small chromosomes, chromosome 22, and then what's the sex of this child or adult? It's got 2 X chromosomes and no Y, so that makes it a female. What else do you notice about this particular map? What do you see on the karyotype? And hopefully you noticed that chromosome number 18 has 3. So it says when there are, trisomies and monosomies means there are 3 of one chromosome. That's a trisomy. That's what we see in this image. Or 1 of one chromosome. That would be a monosomy if there was just one chromosome, where there should be pairs of chromosomes. Trisomies have 47 chromosomes total. Monosomies have 45 chromosomes total for a human. There's 3 traits that are 3 syndromes that I want you to be aware of. We'll learn more genetic traits coming up, but not yet. Okay, so the first one is Down syndrome, and I want you to study the karyotype and see if you can figure out what's going on with Down syndrome. Hopefully you see the arrow, it's kind of obvious. This is called another name for Down syndrome. So Down syndrome is named after the doctor who first sort of noticed these similar symptoms. And so his name was Dr. Down. And so Down's syndrome is trisomy 21. That means that there are 3 of chromosome 21. If there were 3 of chromosome 18 or 3 of chromosome 1, it'd be a totally different syndrome. It is one of the few survivable trisomies. And that's because the 21st chromosome is super, super small. So having an extra one doesn't bring a whole lot of extra DNA into the picture. But trisomies of most of the other chromosomes lead to severe birth defects and usual miscarriage or death. So it is a survivable one. Down syndrome, as many of you know, leads to mental disability, heart defects. You might not be aware of that. And then certain physical characteristics that are very characteristic. So you recognize them. Slanting eyes, flat forehead and back of the head, low set ears. There's a whole bunch of others. Their hands are affected. So their hands look different than other people's hands. Those are all symptoms in that case of Down syndrome. Right, the next one that we want you to know about is called Klinefelter Syndrome. Klinefelter Syndrome are males. They're born males, and in every way, shape, or form, they are physically, in terms of biology, they are males. However, they were born with an extra X chromosome. So they are normal until puberty. And then once they hit puberty, that extra X chromosome leads to too much estrogen. And estrogen is what causes secondary female sex characteristics. So these are boys that have a penis. They don't have a vagina. They don't have a uterus. They don't have, the things that allow women to give birth. But estrogen causes their faces to not develop hair, their voices don't deepen, they develop breasts, which is really socially so difficult for these boys because it's not something that they are asking for specifically. So breast development, no lowered voice, no facial hair, often infertile. And the other thing I'll mention is it is incredibly common. So Klinefelter's is 1 in 500, they estimate. And so at our high school, we would guess we would have 1 to 2 Klinefelter's boys at any time And most of the time, the boys have not been diagnosed. They don't know why they look the way they look. And then just taking a look at the karyotype, so we see that this boy is an XXY, so there's his Y chromosome. That's the Y chromosome is what gives him his genitalia. The X chromosomes are what cause the breast to develop later on in life. And then the other thing that you should notice is that there's also trisomy of chromosome 21. So this is a child that has Down syndrome and is Klinefelter's at the same time. All right, and then the last one that we want you to know about is called Turner syndrome. Turner syndrome is actually only affects girls. They are females that are XO, and when the O, we see the O, that means empty set. So they're missing their second chromosome, their second X chromosome. So you can see here that she's only got one X chromosome. The symptoms are fairly mild. There are certain things that are an issue. They have a characteristic, what they call webbed neck. It sounds, my students are always like, was it webbed neck? And it's nothing. Like you wouldn't even notice. It's just their necks are a little bit wider than an average person's neck is. The one big issue for these girls is that they are infertile. There are a few other genetic issues that come to play with Turner syndrome, but for the most part, they are completely healthy and normal. All right, I want you to take a moment now that you know something about karyotypes, and I want you to study this karyotype. And I'm hoping that you're like, what the heck is happening here? See if you can guess what happened. Okay, so this particular genetic disorder is called a triploidy, and unfortunately for humans, it is always a fatal condition. When triploidy occurs, it usually ends in a miscarriage almost immediately. So within 6, I can't remember the average number of weeks they think that this miscarriage occurs. So it is not a survivable condition. It is due to one egg and 2 sperm fertilizing it simultaneously. So remember that each gamete has 23 chromosomes. So 23 plus 23 plus 23 makes 69. So these are zygotes that have 69 chromosomes. And for humans, it's not a survivable condition. However, take a look at these karyotypes because it's super interesting. Here's a whole bunch of ones that have what's called polyploidy. Polyploidy just means more than 2, so 3 or more chromosomes. And there are some organisms for which this is the norm. We see a high incidence of this in plants, in frogs, in fish. There are other animals. There are advantages. And commercially, I'll just mention that humans have realized that polyploid organisms tend to be healthier. So if we're going to have fish, if we're going to raise fish or we're going to raise certain plants, they have improved growth. They have improved pathogen resistance. They are infertile, and that's actually helpful for the farmers or the people who are growing these species. Because then it allows them to not experience recombination of their genetics. And so once they have a good variety, they won't reproduce with themselves or with others. So just trust me that that's a good thing. Fascinatingly, let me get my picture out of the way here. Fascinatingly, polyploidy in species that can tolerate it, humans can't, but in species that can, it appears to prevent the accumulation of recessive mutations. So in future generations, leading to hybrid vigor. What that means is that the organisms that are polyploid are healthier and stronger and better able to handle, and they're less impacted by mutations. See if you can follow that. It's very similar to the idea of how females are somewhat resistant to mutations on the X chromosome because they have a backup X chromosome. So if I look at this number 5 right here, if I look at these, if this chromosome right here has a bad mutation in it, this organism has 3 other chromosomes that it can count on, and it doesn't have to use that 5th chromosome that has that recessive mutation. So it gives them an advantage, and that's called polyploidy. Humans, no. All right, we're almost to the end. There's not much on this. So now we want to talk about chromosomal mutations themselves. So what we have talked about on this set of notes, we've talked about nondisjunction, where we get the wrong number of chromosomes. And we've talked about then monosomes, trisomies, triploidies, polyploidies. That's the wrong number. But there's actually things that can happen to the chromosomes themselves. Now, in our next unit, we'll talk about tiny, tiny, tiny things that can go wrong within a gene. This is the chromosome itself. So there's 4 main types of chromosomal rearrangements, things that can go wrong with a chromosome. Number 1, you can have a deletion. So take a look right here. Do you see the D is missing? So that's an example of a deletion. Number 2, we can have such a thing as a duplication, where a section of a chromosome gets copied more than once. And so that allows it to have too many of the particular gene, and that has different impacts depending on the situation. Inversion, it's a good word. It just means inverted. It flipped over. So look at how it's backwards. In the original chromosome, it's B, C, D, and now it flips over. And now in the second chromosome, it is D, C, B. It's in the wrong order. That's an inversion. And then the last one that we're going to talk about is called translocation. Translocations are a big deal. We'll talk about them more a little bit later on. But translocation is when one chromosome breaks away, a part of a chromosome breaks away, and then it reattaches onto a different chromosome. That's called a trans. Trans means to move, and then location. So it moves locations. All right, and then I'll just show you this quickly. Some rearrangements, some of these chromosomal mutations are detrimental. They can be deadly, they can cause diseases or disorders, genetic disorders, but sometimes they're what is called silent, which means that they don't have any impact at all, and the person who has these effects has no idea. Sometimes the only way we know that we have some of these chromosomal rearrangements is if it shows up in our offspring and causes birth defects in our offspring. So just take a look. In this case, there's a translocation. Normal chromosome 9 breaks off, and then it attaches. They have sort of a reciprocal translocation where they both break and reattach, and that can lead to certain types of leukemia. And then this is kind of hard to see. There's a karyotype under here. It's kind of a cartooned karyotype. And then everywhere you see a little colored banner. That's where there was a translocation from one chromosome to whatever one it's reaching over to. And you can see the names of some of the different disorders that have been identified as translocations: schizophrenia, leukemia, lymphoma, leukemia, Down syndrome. Anyway, so that's it, ladies and gentlemen. Please let me know if you have any questions.